This tutorial will introduce you to the Structure Function Linkage Database, or SFLD, which links enzyme sequence and structural information to specific functional information. I'll show you how information in the SFLD is organized and some of the basic functionality available using the radical SAM superfamily, a diverse group of evolutionarily related enzymes that use a conserved 3-cysteine motif to bind an iron-sulfur cluster and activate S-adenosylmethionine, as an example. There are a couple of ways to get into the database. I'm going to start by browsing through the available superfamilies, so I'll click on this Browse by Superfamily link at the top of the page. I get a list of the superfamilies in the database divided into two sections. The core SFLD contains our most extensively curated superfamilies, and the extended SFLD contains superfamilies that are less well curated. In this tutorial, we'll be looking at the radical SAM superfamily, which is part of the core SFLD. Sequences in each SFLD superfamily are organized hierarchically based on sequence, structure, and function. At the top level of the hierarchy is an enzyme superfamily. The next level of the hierarchy is a subgroup. In the radical SAM superfamily, for example, we have 49 different subgroups which have been defined based on overall sequence similarity so that enzymes in each subgroup are more similar to each other than they are to other enzymes in the superfamily. The next level down in the hierarchy is family, where again, enzymes in a family have more sequence similarity to each other than to other sequences within the parent subgroup. But more importantly, they're thought to catalyze the same overall reaction based on the same mechanism. We currently have 93 families defined for radical SAM. The superfamily names in this table are clickable links. Clicking on any superfamily name will take you to the corresponding superfamily page, so I'll click on radical SAM to go to the radical SAM superfamily page. At the top of the superfamily view, we have some basic summary information about the superfamily. Because sequences contain multiple domains that may be different folds with different catalytic functions, we assign only the region of a sequence with the appropriate fold and function to a superfamily. We call this region a functional domain. So here we have listed the total number of functional domains in the superfamily. These are subdivided into those that have been assigned to a family and those with no family assignment. The functional domains that have been assigned to a family are further subdivided into those that share 100% of the family-specific catalytic residues identified by curators and those that don't, which may indicate that they're outliers. These numbers are all clickable links, and clicking on a link will take you to a table with more detailed information about the corresponding functional domains. We also list the number of UniProt knowledge base identifiers and NCBI-GI numbers that correspond to these functional domains. And finally, we list the number of structures and reactions in the superfamily. To the left of this summary table, you see an example superfamily structure. And just under that, we have a text description of the superfamily. Clicking the References tab will give you a few literature references for further reading. The Cofactors tab shows you the cofactors used by all enzymes in the superfamily. And the Partial Reactions tab shows you the chemical capability shared by radical SAM superfamily members. In this next set of tabs, you can access the curated alignment for the group and also some download tools. We'll look at those in more detail in parts 2 and 3 of this tutorial series. Skipping down to the bottom, we have a tabular view of the way the sequences and structures are distributed between subgroups and families. Subgroups are shown in blue and families in yellow. The column with the T heading lists the total number of functional domains in the group. The column with the K heading lists a count of those functional domains with family assignments, in other words, those where we think we know the function. The column with the C heading lists the number of functional domains with 100% conservation of the group-specific catalytic residues. The column with the U heading lists the number of functional domains without family assignments, in other words, those where we don't know the function. And the column with the S heading lists the number of structures available from the protein data bank. And all of the subgroup names, family names, EC numbers, reaction names, functional domain, and structure counts are clickable links. So if you want more detail about anything, you can click on the associated link. That's your very brief introduction to the SFLD. If you'd like to explore the database in more detail, see Part 2 of this tutorial series, where we'll explore how to use the SFLD search and alignment tools to make hypotheses about the function of an uncharacterized enzyme.